This is a video to recap the uses and the stages of PCR, and we'll also look at how to complete a PCR calculation. So PCR, or polymerase chain reaction, is the process by which DNA is amplified, and this just means to make lots of copies of the DNA. I always think of it like an artificial form of DNA replication, so it's happening outside of the body in a lab. And PCR is really helpful for three things. So we use PCR in solving crimes. This means if you find maybe a small amount of DNA at the crime scene, you can amplify it and make lots of copies of it. And paternity testing, so settling paternity suits, finding out who the father is, and also diagnosing genetic disorders. And we're going to look at what happens at each of the three stages involved in PCR. Okay, so PCR is a cycle of heating and cooling, and we have three stages that we need to know about. We need to be able to say what temperature the DNA is at when these processes are happening, and we need to be able to describe what is happening at each stage. So if we look at this first diagram, we can see that we have our original section of DNA. So this is the section, or this is the part of the DNA that we want to replicate. And we have some free DNA nucleotides and we have some primers, and all of these will be going into our PCR machine. And for stage one, we can see that we have separated those strands, and the way that we do that is we heat the DNA up to 92 to 98 degrees Celsius, so really high temperatures here. And what that allows us to do is break those weak hydrogen bonds between the DNA strands and separate the DNA strands. So 92 to 98 degrees, and we're breaking the hydrogen bonds and separating the strands. Now what this leaves is two completely separated complementary strands of DNA that we can start to replicate. Okay, so for stage two, what we are doing is we are cooling down the DNA, and what this is allowing to happen are these primers to bind to their complementary specific target sequence on the DNA. So remember, primers are always binding to the 3' end of the DNA, but because we have completely separated these strands in stage one, it means that we can just replicate each of the strands continuously. We don't have a lagging strand like we do in regular DNA replication. And we need to specify that they are complementary because they will be joined together by those complementary base pairings. Okay, so now we're going to start replicating this DNA, and for that we need DNA polymerase. And remember, DNA polymerase is adding DNA nucleotides to the 3' end of the new strand that is forming. So DNA polymerase is just going to be moving along both of these strands continuously and adding these DNA nucleotides to their complementary bases. So for stage three, we are heating the DNA back up to 70 to 80 degrees. And the reason that we do that is it is allowing optimum conditions for this enzyme, for the DNA polymerase to replicate that region of DNA. We also need to specify that the DNA polymerase is heat tolerant because it's going to have to be present during stage one when we heat that DNA to 92 to 98 degrees. Okay, so there's 5 things that we need to add to our PCR machine in order to carry out this process. We have our DNA template, and remember we have completely separated these strands here, so we've got our DNA template that can be replicated. We need our primers, which remember are going to bind to that 3' end, they're going to bind to that complementary specific target sequence on the DNA. We need DNA polymerase, which again is going to add those DNA nucleotides to the 3' end of the new strand that is forming. We need our DNA nucleotides, and we also need ATP. This is a process that requires energy. Okay, and this is the kind of way that it could appear in a past paper question. So we can see that heating and cooling process, and we can see that we've got stage X, stage Y, and stage Z, and we would need to be able to describe what is happening at each of those stages. You need to be really familiar with the wording here. They are very exact with the wording that they want. So again, for stage X, 92 to 98 degrees, breaking the hydrogen bonds and separating the strands. Stage 2, we can see that it is cooling. So we're cooling it to 50 to 65 degrees and we're allowing the primers to bind to their complementary specific target sequence on the DNA. We need to specify that it is on the DNA. And then we're heating it back up to 70 to 80 degrees, and this is allowing optimum conditions for the heat-tolerant DNA polymerase to replicate the region of DNA. And remember that it needs to be heat-tolerant because it is present in stage 1 when we're heating the DNA to 92 to 98 degrees. So PCR calculations are common questions that can come up in your exam. And you might be asked to work out how many copies of DNA will be made within a certain period of time, or sometimes you have to work out how long would it take to produce a certain number of copies. And we're being asked here, if there were 500 molecules of DNA at the start, predict how many copies there will be after 20 minutes. Now, if we look at the graph, we can see that we have this repeated cycle of heating and cooling. And what we're trying to work out is where is the pattern starting to repeat itself. That's going to allow us to work out how long is one cycle of PCR. And I always find it easiest to look for the double drop. So there's my first drop, there's my second drop. That is where the cycle starts to repeat. So 2 to 6 minutes, one cycle is 4 minutes. And it's telling us that we start with 500 copies of DNA. Now, because we are completely replicating that DNA every cycle, after 4 minutes, we're going to have 1,000 copies of DNA. After 8 minutes, we're going to have 2,000 copies. After 12 minutes, we're going to have 4,000 copies. After 16 minutes, we're going to have 8,000 copies. And after 20 minutes, we're going to have 16,000 copies. And that is what we were being asked. How many molecules of DNA would we have after 20 minutes? It would be 16,000.